Welcome back to the Turd Nerd Check-In series, where we've been learning a lot about how manure and other agricultural waste products can positively impact soil health. Today, we're going to talk about soil microbiology. We've talked about soil microbiology a little bit when we talked about soil respiration, the CO2 that's being released by microbes. And we also talked about soil microbiology when we talked about carbon degradation, because soil microbiology is a big part of that. We touched on a little bit when we talked about soil bugs, soil arthropods, because those are a part of that larger soil sort of food web and ecosystem. So today we're going to talk about measuring a little bit more directly number and variety of microbes that are in the soil. So why this is important? We've kind of mentioned how important soil microbes are for all the different functions that they have. They are the ones who are responsible for cycling carbon, so sequestering carbon, degrading plant materials that are left on the soil after harvest. They're enriching the soil. They're part of the nitrogen cycling system, which is how plants ultimately get their fertilizer. They're really important for keeping nutrients in the field because they can transform something into an organic form temporarily and then back into a plant available form. And that is important because an organic form is less likely to run off in water or get down into the groundwater. Organic forms are a little bit more sticky throughout the season rather than a big bunch of inorganic fertilizer at one point, a lot of which is going to get lost and not available to the plants. So that's an inefficient application, but where it gets lost, it usually becomes a problem if it gets into groundwater or surface water. We want to know what all of the different species that are in the soil and how they fit into each of these different cycles. So there isn't one cell, one type of species that is going to do all of those things that I was mentioning. They're not all involved in all of those cycles. They don't all have the same, what we call niche within the ecosystem. They all have different functions. And so if we want to see not just looking for the effect in one space, but are we building a broad and healthy, because it is broad and sustainable soil ecosystem, we want to look at a variety of the functional types of bacteria that are in the soil. So to do this, we're basically going to feed bacteria that we pick up from the soil a variety of different foodstuffs that are food for them, not food for us, taking the form of what we call substrates. Each of these different kinds of substrates are different sort of molecular forms that are only going to be used by certain types of bacteria. And if they're all being used, then we have a feel for that we have a very diverse species set. And if we're only seeing a little bit of use in some categories, then we don't have the same diversity. But we're going to measure how much of the food stuff that we give them is depleted. There are millions of bacteria, even in a very small sample of soil, and that would overwhelm our testing process. So we would receive so much information that it is not easy for us to distinguish what we're seeing. So what we're going to do is we're going to do what's called serial dilution, where we will take a little bit of soil into some amount of a diluent. It could be water, it could be some kind of medium, and then we'll take a little bit of that mixture into the same amount of diluent, and then we'll do that again. With each one, we're reducing the concentration from the original sample by a multiplier. So once we've made our dilutions, we're going to put a little bit of our dilution into each well of a microplate, and each of the microplate wells, and there are 96 of them, have different substrates. And how we're going to measure that is using photospectrophotometer. What we do is we put the microplate with all the wells, with the bacteria and the substrates into a machine that passes light through it. And then how the light is changed as it passes through the material depends on how it is interacting with the particles in that solution. And so if we use the right kind of initial light and we're looking for a specific indicator,
color,
then we can see how intense that indicator is in our solution. And that will tell us how much food has been eaten. And you can see that in these pictures, we have a large variety of intensity in the bacteria that we see. And that is how we measure functional microbial diversity here in the Schmidt lab. So that's what we're up to this time. Tune in again next time for more Turd Nerd Check-In.